It is my privilege to introduce our speaker today, Dr. Collin Anderson of the University of Sydney. He's a lecturer in the School of Medical Sciences and the School of Biomedical Engineering at the University of Sydney. He earned his Bachelor of Science in Biomedical Engineering at Johns Hopkins University and his PhD in Bioengineering at the University of Utah. Well, afterwards, he completed a postdoctoral fellowship in neurology at the University of Utah prior to starting his own research group at the University of Sydney in 2023. Dr. Anderson's research interests revolve around disorders affecting the cerebellum and basal ganglia with multifaceted translational approaches that encompass neural engineering and molecular and genetic techniques. So, Dr. Anderson, the floor is yours.
Great, thanks so much. Let me share my screen here. Great. Can everyone see that okay?
Yes, we can.
Perfect. That sounds good. Apologies if I seem a little sleepy or if I yawn. It's 3 o'clock in the morning here. And that's not a typical time for me to be awake, but I didn't want to pass up the opportunity to give this talk. So thanks for the introduction. So I'm a lecturer in the School of Medical Sciences, primarily with an affiliation with Biomedical Engineering, and I work in the Brain and Mind Center at the University of Sydney. So being a lecturer is like the equivalent of an assistant professor on the UK-based system that we have here. So I'm in a pretty typical position that's split between teaching and research. All the data that I'm going to talk about today were collected prior to leaving Utah almost exactly a year ago. We've been continuing some molecular analyses out there, having some colleagues finish some collection up for me, and then we've been doing some additional analysis. We're excited to have some new data to share on this story. So kind of what I'm going to talk about today starts with the 2017 postdoctoral fellowship that I got from NAF, and then continues on through work that they funded in a young investigator grant in 2020. Here in Australia, we like to acknowledge the country and the traditional custodians of the land that we're presenting on or that we did the work on as we begin to talk. So today I'm on the land of the Gadigal people of the Eora Nation. It's unceded land that's been part of the kind of traditional ownership of Indigenous Australians for the last 65,000 years. And I also want to acknowledge the traditional owners of the land on which I did this work in Utah. So disclosures, I have funding sources up here. I want to highlight these two awards from the National Ataxia Foundation, which really enabled all of this. So I've had the opportunity to do work in a handful of really beautiful places now. So you can see why it was easy to stay in Salt Lake City for both my PhD and my postdoc. And then I've kind of traded in the mountains for the ocean here in Sydney. I've really loved my first year out here. So I like to start with a little primer on the little brain, as I call it. I don't think that's actually that important for the audience today. I would assume that everyone in the audience kind of knows these things, but just kind of briefly, we're going to be talking a lot about Purkinje cells today. Purkinje cells being very important as this output from the cerebellar cortex, and then synapsing onto the deep cerebellar nuclei. And I think that I just want to draw attention to this, this kind of repeated, very organized organization within the cerebellum. So I can't give a cerebellum talk without showing a video of my cat Phineas, just to give an example of what cerebellar disorders look like. So shortly after I joined Stefan's lab, Stefan Polst in the University of Utah, I ended up adopting a cat with cerebellar hypoplasia, which just means that his cerebellum was underdeveloped at birth. So you can see he has a very ataxic, wobbly gait. He exhibits several different types of pretty stereotypical cerebellar tremor. So that's him when he was a few months old. Because his cerebellum was underdeveloped, hasn't really gotten better or worse much over time. So he still kind of looks like this now to this date. If you find yourself bored during my talk, there are actually quite a lot of articles that have been written about him recently, which is kind of funny. Just a week ago, we were in the American Academy of Neurology, Brain and Life magazine. We had an article in Sydney Morning Herald earlier this year, and something from Newsweek like a year ago, which is kind of funny. So if you get bored of hearing me talk, that might be more interesting. So when we study cerebellar dysfunction preclinically, when I joined Stefan's lab, I was really interested in applying neuromodulation, so something like deep brain stimulation to the cerebellum to try to treat cerebellar tremor and ataxia. And so I was interested in using a model that had very exaggerated symptoms. And he immediately suggested the shaker rat, just a spontaneously occurring natural mutant model. The gene was not known at the time. And what we did know at the time was that if we compared shaker rats on the bottom here in these two at 2 and 6 months versus wild type animals at 2 and 6 months, what we could see was that shaker rats had severe degeneration of Purkinje cells in every lobule outside of lobule 10. This heavy loss of Purkinje cells led to a very exaggerated and predictable progressive ataxia with a tremor phenotype as well. So I thought that this was a good model to work with, because if we had such severe symptoms to start out, it would be easy to quantify that. And then basically we have a large difference between wild type and shaker rats that we could attempt therapeutic procedures in. So in a degenerating cerebellum, you're losing these Purkinje cells, which provide a tonic inhibitory pulse to the deep cerebellar nuclei. And then basically they can disinhibit the deep cerebellum when they're intending to make coordinated movement. And so basically you losing these Purkinje cells means that you lose the ability to fluctuate these inputs to the DCN. So we thought in the absence of the Purkinje cells, maybe the best thing that we could do neuromodulation-wise would be to go immediately downstream of that and see if we could modulate the network in some way that would restore some degree of coordination or reduce tremor. Being an engineer and coming into this, we thought it was really important to be able to quantify motor symptoms directly. There's a lot of utility with a lot of different types of measuring cerebellar motor dysfunction with things like rotarod. It's pretty common, but I wanted to be able to measure these things directly. So measuring how straight the gait was, kind of to measure ataxia, and then directly quantifying tremor. So in terms of tremor, we just did a pretty basic power spectral analysis where basically we're quantifying in the context of wild type animals and then quantifying an area under the curve as it's separated from wild type animals. So you can see here, this particular animal has a peak at 5 hertz. And this is representative of a 5 hertz tremor in the animal. In terms of measuring ataxia, what we did was we took all of the kind of rapid movements that the animal would make and then automatically detected those. And then we quantified in all of these basically a ratio of the total distance traveled compared to the net displacement. So basically a higher number would mean more ataxia. So just as an aside, we're doing all this with what's called forced plate actometry. It's not really a new idea. It first came out over 20 years ago now. But in my group here, we're actually working on some very low cost budget options that we're trying to design it with as many 3D printable parts as possible. Basically the goal being that we can publish this and it can be manufactured at any university with a just basic fab lab. Complete with open source software. So this is something we're hoping to get out by the end of this year now. Here's something we did with this recently, just in the context of using a naive Bayesian classifier to detect grooming behaviors in a mouse model of Tourette syndrome, which I thought was pretty cool. Worked super well. So in the shaker rat, before we started getting into therapeutics, we wanted to make sure that we could fully characterize and quantify the symptoms in this model. So in terms of tremor, what we found was that Finn has just a beautiful several examples of tremor here that you can see several different types of this cerebellar tremor. So we're measuring kind of across all these different types in the shaker rat. We're a bit agnostic as to what type of tremor it is specifically, but just in terms of that power spectral increase in movement in kind of a range that's situated around that 5 hertz kind of median level. So what we find in the shaker rat is they go from asymptomatic to having a pretty strong tremor in a short period of time. This graph kind of appears to be bimodal, but really this is, we have some litter effects in this particular first cohort. We had a few stragglers that just took a little bit longer to onset. Typically speaking, once the animal has a tremor, it comes on pretty strong and it actually dissipates to some degree over time as it kind of evolves and becomes more ataxic. In terms of the ataxia, again, I like to use Finn here. You can see this truncal sway as his body goes back and forth as he travels to his food bowl. And so what you can see is, in contrast to the tremor, the ataxia develops gradually over the span of several months in this model. And it's kind of mirroring the loss of Purkinje cells that we see in this particular model, which tends to start at about 7 weeks of age, continues to maybe around 17 weeks or so. And so we have an incoordination that's fairly in hand with that. So we started off by wanting to just use something akin to deep brain stimulation to see if we could reduce motor symptoms in these animals. So DBS has been used upwards of 200,000 times. That's actually probably a bit of an outdated number. I think it's over that now. Typically in DBS for Parkinsonism or essential tremor, it's a high frequency stimulation pulse of 130, 180 hertz, somewhere in that window. Based on my PhD data, we like to think of it as functioning like a lesion effect. And so we would call it an informational lesion. So essentially, if you have an overcoupling of different neurons in different nuclei, DBS can kind of break that apart by just regularizing the firing patterns when it's applied at a high frequency. So you can do it open loop or a closed loop. It's being used for a number of different conditions already. But there's a big question of how exactly it works. So like I said, I like to think of it as an informational lesion. This is something we published in 2016. This was my first authorship of my PhD. So what we were doing was we were measuring downstream of subthalamic nucleus stimulation in the substantia nigra pars reticulata, the most important output from the rodent basal ganglia, and then its receiving area in the thalamus. So we're doing simultaneous multi-channel recordings of action potentials in each of those. And what we can do is we can take the firing patterns in each. So we can look at cross spike intervals. So if you have at time 0 is when the downstream neuron has just fired an action potential. The cross spike interval is how long it's been since the upstream neuron has last fired. The inner spike interval is how long it is until that particular neuron fires again. And so what we find is if we're measuring what we call quantitative information, basically what we can say is in this example of a highly informative pair, every time that there has been a long cross spike interval and neuron A hasn't fired for a long time before B fires, you're more likely to have a shorter inner spike interval. And if it's been a short time since neuron A is fired, you're more likely to have a long inner spike interval. So basically what that means is that having a priori knowledge about neuron A is predictive about what's going to happen in the future about neuron B. And basically what we're able to show is that in a Parkinsonian brain, what we found was that there's a greater number of neurons that are coupled than you would find in the healthy scenario. And then in measuring across these same exact neurons, say 5-10 minutes later with deep brain stimulation, what we find is that when it's effective, it's restoring that information channel independence. So that's kind of how we like to think about it working. We call it an informational lesion. So based on that, how can we apply this kind of knowledge to target deep brain stimulation to the deep cerebellum? Now, the problem here is kind of different from in Parkinson's disease, where in the absence of Purkinje cells, what we're thinking about is that we're missing an important input to the deep cerebellar nuclei. So then the question is, rather than trying to decrease the amount of informational throughput, what can we do that might enhance any remaining throughput rather than blocking signals? There was an idea that kind of came out of Ken Baker and Andre Machado's work, which was actually targeting low frequency stimulation to the deep cerebellum for stroke recovery in terms of potentially enhancing throughput of remaining informational channels. So we decided to take that. We tested across a number of different frequencies. And I'm going to gloss through this pretty quickly because this isn't the primary focus today, but I think it's a nice lead into the gene therapy work, just showing the techniques. So basically what we found was when we were stimulating at any frequency outside of the tremor physiologic range, we couldn't really worsen the tremor unless we were stimulating at a tremor frequency. But these low frequency stimulations, so say from 10 to 40 hertz, we could significantly improve the tremor. It's interesting that typically high frequency stimulation is associated with improvements in tremor. But in this particular case, what we were finding was that we had very, very low side effect thresholds at high frequency. So basically we weren't able to stimulate it with enough amplitude to decrease the tremor. On the other hand, with ataxia, what we found was that low frequency stimulation was capable of reducing ataxia by a modest degree, but high frequency stimulation was actually worsening the ataxia, which kind of makes sense if it's functioning as an informational lesion. But so basically in the end, we're able to reduce tremor by about 50% and ataxia by 25 to 30% with low frequency stimulation, which we thought was really promising. And as I said, the thresholds for side effects that we were finding were quite a bit lower at these high frequencies than they were at low frequencies. So we're trying to stimulate up here. We can get a lot more amplitude and we get more benefit in terms of tremor. And one kind of nice sanity check that we were able to find was that we had one particular animal where with the bilateral stimulation, the electrodes were far too, both electrodes were far too medial and anterior. And in that case, we were not able to generate any side effects with stimulation and also didn't have a therapeutic benefit. So it kind of gave us some indication that we were targeting well in the other animals. So this work is still ongoing. Roy Silito's group has taken this well beyond what we've done. And actually, it's gone into a small number of human patients in Brazil. I believe there have been 8 implanted down there so far, and 7 of them have seen some fairly comparable degree of improvement compared to what we saw in the rats. So this is exciting. But the question kind of becomes, this is a genetic model. Clearly, it's spontaneous. So if we could figure out what the genetics are in this model, is neuromodulation always going to be the best that we can do? So basically, as we were working on this, working with Patty Figueroa, the lab manager in Stefan's lab, and kind of, we like to call her a mouse geneticist, she had identified SLC9A6 as a potential candidate gene that could be underlying the shaker phenotype. So I'm not going to go into too much of her work there, but basically we kind of homed in on this as the likely candidate gene right at the distal end of the X chromosome. And what we found was, so SLC9A6 being a solute carrier is creating this NHE6 protein. It's a sodium hydrogen exchanger number 6, hence the name. And so what we found was in a heterozygous female, on average, we'd have about 50% of that protein level. In a shaker rat, we had none. So this was good indication. We had a good antibody that could work here in Western blotting. What was particularly promising was that loss of NHE6 function causes an ataxic disorder in humans called Christianson syndrome. So it's an X-linked recessive condition, which seemed to mirror what was going on in the rats. So we decided to try to basically determine if we had the right gene through functional complementation studies, which kind of at the same time, we're using AAVs, so viral vectors, to replace that missing gene in Purkinje cells, kind of that can it's not only a functional complementation study, but it can also kind of lead us down a path towards therapeutics. So we were predicting that the shaker symptoms were caused by a frame shifting mutation in SLC9A6, which results in a very early stop codon and total loss of function associated with Christianson syndrome. This is an MRI from Eric Morrow's group at Brown. This is the same patient at, I believe, 4 and 14 years of age. So you can see this severe degeneration of the cerebellum there, which is associated with ataxia. So what happens without a functional SLC96 gene? So it's expressed in all neurons, but it's very highly expressed in the cerebellum, the hippocampus, and the forebrain. And consequently, the symptoms that emerge are ataxia, severe cognitive deficits, epilepsy, and autistic features, among a number of other things. What's interesting is we really only thought about this as a rat model of Purkinje cell loss and ataxia, but thinking about just looking at a rat in a cage, it's kind of hard to determine that they have cognitive deficits without running a pretty serious battery of tests. And in terms of the epilepsy, maybe we'd seen or heard of a seizure happening here and there, but not that many of them. We'll get into that in a little bit. But basically, what we wanted to determine was, do we have the right gene here? And we can figure that out by basically replacing the gene in Purkinje cells prior to their loss using an adeno-associated virus. So it's just a small non-replicating virus, basically little more than a protein shell and a small single-stranded viral genome. And so basically what we started with was, you can see in blue here is just a DAPI stain, green is green fluorescent protein. So we started with using an L7 promoter that specifically targets Purkinje cells, and then just basically administering a virus through directly into the ventricle. What we could find was that basically we get specific targeting in Purkinje cells. So this is just a couple different concentrations of virus that we were administering, a relatively modest and then a slightly higher concentration. You can see that in both cases, we get specific uptake in the Purkinje cells. So at that point, we decided to replace the transgene with the SLC9A6, the rat copy, with a fused GFP tag at the end of it. And we were doing this all using the PHP EB capsid that had been engineered at Caltech a year or two prior. So when we put this in, what we found was that first and foremost, very excitingly, the motor performance was rescued through use of this AAV. So across all this work, we have 4 different groups. So as a reminder, we're quantifying straightness of gait here, so you can see a few representative traces. And what I've shown first is just the ataxia at 25 weeks of age. This is actually statistically the most conservative time point of this study. It's the last time point, and it's the one where the stats are actually the weakest. But still, we have a very significant reduction in ataxia between shaker rats administered a control vector that's just an L7 GFP, and those administered the SLC9A6 GFP vector. And we actually have a handful of animals that don't look that far off from wild type animals here, which is pretty exciting. So in the wild type category, we had uninjected wild types, and then wild types administered that control L7 GFP vector just to make sure that the administration of the vector itself wasn't causing any changes in gait, which it was not. And what was cool is if we automatically identify all the rapid movements that the animals made during these weekly motor recordings, so this is at 25 weeks, every single dot corresponds to a different movement that was made during that half an hour when they're uncued in the chamber. What you can see is that 4 of the 8 animals here in this treated category never make a single movement that's severely ataxic. So that's half of our cohort at the final time point when the animals should all be pretty severely ataxic without treatment. Half of the animals in the cohort are never making a severely ataxic movement. So this was really exciting for us to see. So this is published. We have some additional data that didn't make it into the first paper that's part of an ongoing in preparation manuscript at the moment. So what we've actually done is we've quantified this motor performance across, so it's 30 minutes per animal. I believe there's a total of 22 animals in the study. So that's weekly. We have about 660 minutes of motor data across a span of 20 weeks. So it's a massive amount of motor data that's going into this. And basically what you can see is the wild type plus GFP versus the wild type is pretty much the same. You have these untreated shaker rats that are progressing pretty identically to what we found in our previous study that we published in 2019 using the DBS. And then when we have the treated shaker rats with this L7 SLC9A6 GFP replacing just in Purkinje cells, what we find is that we have a much reduced slope, so a slower progression and a much lower peak in terms of that ataxia. So we calculated a cumulative ataxia burden. So this is starting from 10 weeks on, which is kind of the initial symptom point. Obviously, wild type animals, wild type plus GFP, aren't really expressing, they're not exhibiting any ataxia, so these are about 0. Untreated shakers have quite a bit more ataxia than the treated shakers. So we have a couple that aren't doing quite as well, but in general, this we have a reduction in the total symptomatic burden by almost exactly 50%, which is pretty promising. And in terms of, we also wanted to calculate when there was an onset crossing several different thresholds. So a threshold, a ratio of 2.0 in terms of that distance versus displacement is like a very mild ataxia. 2.4 would be kind of moderate to severe, and a 2.8 would be a pretty severe ataxia. 3.0 is really about as serious, as severe as would be averaged over for any animal over, say, the final 5 weeks of the study when they've peaked in that measure. So basically what we've done is we've taken these animals, and not only have we decreased that ataxic burden, and we've heavily dropped the kind of the peak ataxia, but we've also delayed the progression as well. And so if we also look at the maximum ataxia, so kind of the peak ataxia that they display versus the condition, we've dropped that as well, which is really nice to see. Moving on to tremor, it's a bit more complex, actually. It seems like it's perhaps because the tremor kind of peaks and then drops off a bit over time, and we've kind of delayed this onset. Basically, what we find is there's not nearly as much separation as there is in the ataxia at these later time points. But if we run a two-way ANOVA with time as a variable, it's a very, very highly significant decrease in ataxia or in tremor over time. So if you look at the cumulative tremor burden, once again, we have a significant drop-off. These two that didn't do so well, there's actually kind of a very interesting story that I don't really have time to get into today, but as we finish up this manuscript and get it submitted, I would encourage everyone to read that, because I think there's some interesting lessons to be learned about what may have happened in these couple animals. In terms of the tremor onset, this is also pretty interesting that we've certainly delayed the onset of crossing several thresholds. 0.1, so this is a scale where 0 is no tremor, 1 is what we would consider to be a very strong tremor, and 0.5 is already a pretty severe tremor. 1 is about the peak that an animal would at least the average across the cohort would hit. So in terms of each of these thresholds, we have a delay in terms of the age at which that threshold is surpassed, which is really nice to see that delay in tremor. And if we look at the maximum tremor versus condition, we also have a drop-off here as well. So this is really promising. Finally, in terms of these motor performance, what we can say is if we create kind of a total symptomatic burden measure where across the cohort, we have a peak of approximately 1 for tremor and approximately 1 for ataxia, kind of equating these two, what we find is clearly we've decreased the slope of the ataxia, and we've also decreased this amount of tremor. And it seems like we've kind of decreased the slope there as well, too. So this total symptomatic burden is quite a bit lower in the treated animals versus the untreated, which is really promising to see. I think that it's worthwhile to visualize this in terms of that motor performance. So these are animals that are videoed all at the same age of 18 weeks. So this is untreated shaker rats. Basically, you can see there's a pretty severe tremor still, and then there's a fairly high degree of ataxia. So Christianson syndrome being X-linked recessive only occurs in males in humans, but in the context of rats, we can breed a homozygous male with a heterozygous female and end up with homozygous females as well. So either way, it's a total loss of function of NHE6. Functionally, we see pretty similar results between the males and females. Haven't really found any differences there yet. So in all of these cases, I'm showing at least 1 female and 1 male, and I'm trying to make them litter matched as much as possible across groups. So in terms of the animals that have the strong treatment effect, these animals are virtually indistinguishable from a wild type animal. This is at 18 weeks when we would have, I would say, probably 95% penetrance of strong ataxia and tremor by this point. And across the couple animals in here, the best couple from the cohort of 8 that are treated, you can see that there's virtually no symptoms present. And what's actually really nice to see is even the animals that are in this moderate treatment effect. So I believe this was the 4th and 5th, so right down the middle. You can see that while there's some tremor present and some degree of ataxia, and you might see a paw slip here in a second. Yeah, right there. It's quite a bit less severe than the untreated animals. So if this is our moderate case off of the first dose that we attempted in vivo, I think this is really promising. So kind of moving forward from that, after we collected all this motor data, we were interested to see kind of what was going on at the molecular and cellular levels. So we were very surprised when we did our first Western blot, and what we found was the NHE6, on average, we only restored about 10% of it. So this was a bit shocking at first until we were really kind of diving deep into the literature, and we found a case study of a human patient who had a unique mutation that was causing some exon skipping in about 90% of the transcripts. So there was probably a total loss of function in 90% of the transcripts and had about 10% healthy, like wild type NHE6 expression. And that resulted in a very, very mild phenotype, which is kind of similar to what we're seeing when we restored about 10% of cerebellar NHE6 in these animals. What was interesting is that minimal NHE6 restoration yielded significantly greater benefits in terms of cerebellar health markers. So RGS8, calbindin, PCP2, probably things that most of the audience are familiar with. With just that 10% NHE6, we get significantly better outcomes here. What was interesting to see here is that the Purkinje cell loss is only partially mitigated. I think this is a pretty representative example that I'll show down here. You can see in the wild type and the heterozygous animals, where this yellow stain for calbindin, we're not really missing anything other than the tiny little bit that I accidentally chopped off my slice here. In the shaker rats that are just administered the control vector, they've lost basically every Purkinje cell outside of lobule 10, which is resistant here. And what's interesting is in a treated shaker rat, basically, I think this animal might have had 30 or 40% of Purkinje cells preserved outside of that lobule 10. So it's a pretty minimal amount, but this animal still had a fairly high degree of motor improvement, which I think is really nice to see. Now, before I get into the next capsid that we're working on, I just want to touch on, there are obviously non-motor symptoms that are associated with Christianson syndrome as well. So there are cognitive deficits, behavioral differences, these kinds of things. So we actually did a pretty deep dive into this with Marco Bortolato at the University of Utah. So we actually found quite a large degree of cognitive and behavioral deficits here, sensory motor gating deficits, memory recognition deficits, differences in social behaviors, differences in open field, et cetera, et cetera. So these are the kinds of things that as we go into more ubiquitous expression in the future, these are things that we're hoping we can see improvement in as well. And in terms of non-motor symptoms like epilepsy, we did a 60-day long 24/7 EEG and video coupled experiment with Karen Wilcox. And we were able to find that the rats actually were having a small number of tonic-clonic seizures. I show on here, it's not the most pleasant to watch, so just a warning there. But you can see below, we have the EEG of this behavioral seizure that we can see. There we go. So you can see that tonic extension and then this full rotating behavior. What was actually really interesting about this was that we tried to induce seizures by a few different metrics because we figured rather than doing chronic recordings, which are technically challenging and quite expensive and require a massive amount of data analysis, we thought it would be easier if we could just induce them. So we tried something called maximal electroshock seizure induction, probably not thinking about it quite enough at first. There was some very old literature that supported the idea that perhaps it relied on the cerebellum to induce a seizure. So what we actually found was despite the fact that the Shaker rats had spontaneous seizures, the wild-type counterparts did not, we actually had about 2.5 times the threshold to induce a seizure via this method. So we haven't really identified a great way to induce seizures at a lower threshold across multiple tests, but they are at least having spontaneous seizures. They're just kind of rare. So kind of the current directions that we've gone into most recently is developing a gene therapy that would be more translational. So basically what we're looking at is in terms of this Allen Brain Atlas image here, the wild-type NHE expression in a mouse, we were looking at patterning across different promoters and found that the CAG promoter, which is a ubiquitous promoter, had a patterning that was pretty darn close to the kind of endogenous NHE6 expression. So we decided to proceed with a CAG promoter, and we wanted to test a human-specific drug in the Shaker rats. So we're able to take advantage of the very high homology kind of between the rat gene and the human gene. So we're doing this with CAG and then the human SLC96. We're no longer fusing anything to the end of it because we want it to be as translationally relevant as possible. The other difference that we're using now is that instead of using the PHP-EB capsid, which has kind of since been replaced by some newer engineered capsids, we wanted to use something that had already been approved to go into humans and other disorders. AAV9 was a very attractive candidate for a variety of reasons, including that, and also the fact that I believe it's either just gone off patent or it's going off patent this year. So basically, there won't be a massive cost associated with trying to get this into trials in the future. So we took a very large cohort and we had several dozen animals that were administered this AAV9 CAG human SLC9A6. And the first thing that we found that was really promising was compared to untreated Shaker rats. Basically, we have a very clear separation of groups, high degree of significance that when we administer this CAG-based human SLC9A6 vector, we end up with a small but very significant improvement in the expression of NHE6 in the cerebellum. This average is a bit higher than what we were seeing with the L7, but remember with CAG, we're not just hitting Purkinje cells. So say if we have twice as much overall expression in the cerebellum, it might still be a roughly comparable amount in the Purkinje cells. So even though it's driving much higher expression in Purkinje cells than it is in other cell types in the cerebellum, just remember that there are so many more granule cells than there are Purkinje cells that even a small amount there would add up to quite a lot in comparison. What was really nice to see here is we also had fantastic separation in terms of the calbindin expression. So this is off of a homogenized, both of these are off of whole cerebellum homogenized samples and then Western blotted. And we can see that, of course, so these are taken at 14 weeks, so it's a bit earlier. This is just, that's the time point that we had available before I was leaving Utah to start my lab here. So we probably don't have quite as much, like perhaps some of these, it's not fully just therapeutic. I don't think we're protecting all of these Purkinje cells. I think if we went out another 10 weeks, the values are going to be a bit lower. But still, what's nice to see is at this time point, we have a very clear separation across groups and a high degree of significance. In terms of motor performance, interestingly, despite having a clear increase in nearly every single animal except one, we still have a very large degree of variability in terms of that motor performance. Now, again, this is significant at a below 0.001 level. So clearly, this is working most of the time. But it's interesting to see these animals that are not doing better despite having a clear increase in both NHE6 and calbindin. What was nice to see there, though, is that calbindin is actually highly predictive of gait ataxia. So this is on an animal-by-animal basis. Basically, the more calbindin that we have, the straighter the gait is. And so this is with an R value of nearly 0.7 or negative 0.7. What was interesting to see is that NHE6 was also predictive of gait ataxia, but to a lesser degree. This is significant. It's not surprising that it's less predictive because if we're getting NHE6, say, more into granule cells or glia, that's not going to be rescuing Purkinje cells the same way. So while significant, it's less predictive there. What was actually really nice to see as well, though, is that NHE6 was predictive of calbindin. So the more NHE6 we were able to get into the cerebellum, the more calbindin we had there. So this was actually really cool for us to see. So ultimately, at this point, we're kind of homing in on a lead candidate vector using CAG and the human SLC9A6. And ultimately, we're doing longitudinal studies now this year after finally getting the rats re-derived and brought out here, which is quite a large process to get them into Australia. It took, actually, in the end, it's going to be almost a year and a half total of starting the re-derivation until the point that we get them out of quarantine in the coming weeks and can start these experiments. But basically, we're using this lead candidate gene, and we're going to do a longitudinal study rather than a single time point with the CAG. And we're also going to be coupling that with a dose response as well as a timing of dosing based study. So that's kind of the first thing that we're doing there. And we're coupling with non-motor experimentation as well so that we can see kind of the full spectrum of what this is doing. And finally, in the end, we're going to take advantage of the really strong abilities that the University of Sydney has in terms of small animal MRI, which we're really excited to kind of take into this model. So the conclusions that we have here, so going back to the neuromodulation, we think that it could be viable as a modest symptomatic improvement catch-all therapeutic. Personally, I think that it would have the most potential in terms of sporadic ataxias where we don't have a gene that we can target. But we do think that this low-frequency deep cerebellar stimulation could reduce tremor and ataxia when applied in the right situations. And in terms of Christianson syndrome, we think that gene replacement might be able to provide a treatment strategy. We've seen really promising results here in the rat model so far. And what's been really nice to see is that a very minimal amount of gene replacement yields a very substantial degree of benefit, which is great because we can provide smaller doses and perhaps have a significantly decreased risk of toxicity. What was nice to see is that motor function can be relatively protected despite a fairly major amount of neuronal loss. Obviously, there's a lot more work that's still needed to be done. So I want to acknowledge the Pulsed Lab. I was actually the first biomedical engineer or engineer of any kind that Stefan ever took on into his lab. I think it was probably a bit of a gamble for him, and I'm really glad that he did. I think it's paid off nicely. So the rest of these people here are all from Stefan's lab. They were all instrumental in this work. If you didn't see, the human molecular genetics paper that we published last year was actually a triple co-first authorship with myself, Patty Figueroa here, and then Sharon Paul, which is pretty unusual to have that degree of collaboration that we're all first authors on the paper. So Waruni, Dan, and Mondi also contributed very significantly to this work. My partner, Daria, has always been a great collaborator and has collaborated a little bit on the epilepsy work there. She was a postdoc in Karen Wilcox's lab at the time and has now also started her own lab here in Sydney. Marco Bortolato has been a great collaborator on both Christianson syndrome behavioral work and Tourette syndrome work that I've picked up with him as well. And finally, Kajal is a new PhD student of mine that just started a couple months ago and is getting this project off the ground here so that I don't have to do all of the surgeries myself and all the analysis myself in the future. I'm really excited to be working with her. And of course, Finn, who's kind of always making me think about the cerebellum almost 24 hours a day. And the funding sources. like to particularly acknowledge NAF and the RTW Charitable Foundation, who's given me 3 grants to continue this work now. Just got the 3rd one, which is really exciting. So happy to take any questions that anyone might have.
Thank you for such a wonderful talk and an overview of the sheer amount of work that you've done over these years. Just as a
Thanks.
reminder for attendees, the Q&A is now open. So if you'd like to drop your questions in the Q&A window, you can do so at the bottom of your screen. If you would like to ask your question verbally, you can also use the raise hand feature, and we can call on folks and unmute them so you can ask your question to Dr. Anderson. We
And
already
I
have a few.
apologize. I was going
Oh,
to say,
no.
I apologize if I came off as a little bit sluggish. As a reminder, it's 3:45 a.m. right now, so
We'll keep that in mind.
I'm a little tired.
Yeah, not a problem whatsoever. If you could mind just turning off your slide just so that folks are able to see you a little bit better. Fantastic. So we have a first question here. We have an attendee who asks, so rats don't speak, but we know that there are some visual or verbal symptoms that come along with ataxia. Is there anything we can learn from mouse models in terms of the speech-related ataxia symptoms?
Yeah, that's a great question. It's doing vocalization-based studies is a bit challenging in rodent models. So
Hmm.
I have one publication on that in the context of Parkinsonism.
Mm.
The one way that we were able to very clearly collect vocalization data was actually under the context of sexual frustration of a male
Oh.
animal
Mhm.
that was separated from a female animal in estrus. And we were able to get quite a lot of calls that way. We haven't applied that in the context of the shaker rats yet. We are actually quite interested in collecting via another method that we've applied in some Tourette syndrome models in recent years, which is taking very young animals under maternal separation and basically at around a week of age and collecting that way. So that's something that we would like to do. We haven't done that yet, just based on it carries some degree of risk to the pups being rejected afterwards. And by the time we kind of wanted to carry out that study, we didn't have enough animals around that we could actually risk that loss. So it's something that's been on the list of ideas for quite some time, and hopefully we can get that done this year, but it just depends on success in breeding and how quickly the colony grows here.
Of course, that makes a lot of sense. We also have a question from a lay audience member who is interested in terms of how uh, the changes and symptoms you're seeing in rats, how that might translate to human beings. Would the neuromodulation you're speaking of, would this halt symptoms completely? Would it slow it down? Could you speak to that with whatever knowledge that you have available?
Yeah, absolutely. So I think that I want to be cautious about how I
Of
speak about
course.
neuromodulation in the deep cerebellar nuclei. What we've seen is a modest improvement.
Hmm.
I think it's very unrealistic to expect that there's ever going to be a way that you can stimulate downstream from lost Purkinje cells and
Hmm.
restore function anywhere close to 100%.
Mm.
I think there's clear evidence that we can get some serious benefit, but say in Parkinson's patients, in terms of the changes that are going on in the network, it is possible to get very, very strong, nearly normal function through DBS. But because we need kind of a different mechanism for stimulation downstream from lost Purkinje cells, I think modest effect is probably the best that we're likely to ever see. What was the other part of that question again?
I think you got it.
Okay,
It
all right,
was
fair
that
enough.
was, and
Yeah,
then
okay,
we
sure.
have our first verbal question speaker. Give me 1 second, and we're going to uh, promote the speaker so that she is able to unmute. Hi, Sarah. Can you unmute?
Hi, can you hear me?
Yes, we can. The floor is yours.
Okay, thank you. Hi, Anderson. Actually, I'm a researcher. I'm interested in genetic editing, and I have a question. Do you think maybe in the future we can um, replace the damage that happens to people? Like, for example, if someone is already like can't move right now because of their illness, do you think that using stem cells or gene editing, we can actually give them the ability back to move?
Yeah, so I think with AAVs or similar viral vectors, Once the Purkinje cells are degenerated, I don't think that there's anything that you can do there. I will say, in terms of the use of stem cells, I have some colleagues here at the University of Sydney, such as Lachlan Thompson, who are heavily invested in using these kind of approaches in the context of other disorders, such as Parkinsonism. And they've had pretty strong results in that capacity in terms of replacing missing neurons. And I think that that's kind of the dream is that eventually being able to replace Purkinje cells through stem cell based therapeutics. I think that it's quite a bit more challenging in the context of cerebellar disorders, just based on you don't have kind of one nucleus that where you're trying to replace cells. You have this kind of undulating Purkinje cell layer throughout the entire cerebellum. So I think that in terms of like technical ability that's required there, I think it's a much more challenging problem to solve. I kind of expect that at some point somebody's going to figure out how to do that and solve it, but I don't think it's going to be immediately, because I think it's quite a bit more challenging than it is in a lot of other disorders. But I suspect personally that is something that will happen at some point in the future.
Oh, thank you so much.
And thank you for your question. So we're going to go back to the Q&A window. Our next question has a little bit more to do with policy. So feel free to let us know if this is a little bit outside the scope of your expertise. But for deep brain stimulation, the different neuromodulations that you were speaking of, you mentioned that this is already being used for other neurodegenerative disorders. So if this was going to be used for ataxia patients, what kind of regulatory approvals would be needed if it's already being used in other conditions to be moved into ataxia?
I'm far from an expert on that.
That's
The
fair.
cases that I'm aware of where this has been done have all been outside the US or Australia. I'm aware of a number of cases in Brazil. In terms of the US, I've talked to neurosurgeons that basically think it would be possible to do things off-label. I think that, I mean, I'm somebody that feels that it's important, generally speaking, to really make sure that we have things that, like, understand how it's working and have things optimized as much as possible preclinically. So I'm not necessarily of the opinion that it's wise to just jump into throwing this into all kinds of different ataxias, particularly considering that we're not seeing like a smoking gun here of 80–90% improvement. We're quite a bit shy of that. So I don't know. I mean, I think like regulatory wise, it's probably maybe not extremely challenging, but I think that most people are being fairly cautious about it, just to make sure that we really get it right when we do it. Make sure that we select the right cases, because if we select the wrong cases and we don't see much benefit, it could kind of kill the whole thing before it really gets started. So if there is a possibility of more substantial improvement, but we get it wrong because we don't quite understand enough, which is interesting, because in terms of deep brain stimulation, it was really discovered by accident to begin with. And even still, the mechanism, I mean, decades later, is still pretty hotly debated. But in that case, it worked well enough that it's now gotten into a couple 100,000 people. But I think that in general, that's kind of a success story, but not understanding the mechanism well enough just kind of sets it up for not being as successful as it could be, which, yeah, so I don't know. I would exercise caution regardless of regulatory policy there.
No, that makes a whole lot of sense. And then we have a couple of attendees who are asking a similar question. So I'll combine their phrasing together. But most of the work that you shared with us today is done in animal models, and folks are curious what steps would need to be taken to move this from an animal model system to now being tested in humans. What kind of work would need to be done?
So I think that there's a handful of additional experiments that are really going to be critical at this kind of rodent preclinical stage. Nowadays, in terms of getting kind of regulatory approval to get to like an initial clinical trial, it's still pretty common and expected that you would need to undergo safety and toxicity testing in non-human primates. I would say at this point, I would say I have some degree of concerns that just in terms of the ethics there, because I think the field does have a pretty good understanding of what's actually causing safety issues and toxicity, but kind of the FDA requirement is still to do those studies. which is unfortunate because it slows down treatment and unfortunately causes loss of experimentation and loss on non-human primates in a way that I think a lot of us would feel is not really necessary at this point. But regardless, that's the way that things are operating at this point in time. Even though there is some degree of pushback, I think, across some members of the field that think that maybe we shouldn't always be doing that. Regardless, I'll get off my soapbox. But I guess regardless, that's something that will need to happen. That will take a couple years. So realistically, it's anything that's kind of at a similar stage to where we're at. It's going to be a number of years still. I don't think it's realistic that it would be anything under, say, 5 years if everything goes really well, because we have a couple years of rodent testing to do, and then it's going to be some amount of NHP testing that still needs to happen as well. But I think if everything goes successfully there, and I should clarify, the reason that we need that the NHP testing has always been important is that there's not the same degree of toxicity to AAVs in rodents as there is in primates. I think there is a really great study that came out of Beverly Davidson's group, published in I think it was Nature Medicine a couple of years ago, where they were doing kind of a localized injection of an AAV that was designed for SCA1, I believe, directly injected into the deep cerebellum, and they got a surprising degree of toxicity as a result of this localized injection. And I think that does give us this caution of just how we're administering AAVs and the doses that we're using, things like this. So that's kind of why I think that's one of the major reasons why these NHP studies are being done. So yeah.
That makes a lot of sense. And that goes back to the previous point you made about being cautious and making sure we're doing all the correct steps in order to be moving this along in a safe way. And then going back to animal models, we have someone who's curious about how you select the animal models you use to model these different disorders. So why rats?
Yeah. So look, we got into studying Christianson syndrome kind of the opposite way of most people. So there are a few groups that have been doing work in Christianson syndrome for a long time. In studying NHE6, SLC9A6. And so they kind of started with, when these groups were interested in this, they had to develop kind of NHE6 null mice. And there's actually now an NHE6 null rat that's pretty recently been published on as well. So there are a couple different mouse models, and then now there are 2 different rat models. What's interesting is that people have inadvertently been working on Christianson syndrome without knowing it for actually about 30 years now through the shaker rat, which is actually really cool that we discovered that this was a model of Christianson syndrome, because now we've tied 30 years of work and about 20 different papers to Christianson syndrome, and now we can learn quite a lot about Christianson syndrome from this previous work. So from my perspective, we kind of got into working on Christianson syndrome a bit by accident. We thought that we just had a really nice model of cerebellar degeneration, ataxia, and tremor that we were interested in studying. And then as we determined that it was a rat model of Christianson syndrome, and we had collected all this data, we thought, well, this is the one we want to use because this is the one that we're most familiar with and have years and years of motor and molecular and cellular data going into it. In terms of rats versus mice, I mean, we started using a rat for a handful of reasons. First, just basically due to the pretty exaggerated symptoms, but also if we're trying to do neuromodulation, at least for me as a kind of rodent neurosurgeon, I feel a lot more comfortable doing it in a rat just because the deep brain targets are quite a lot larger. So I have a much higher chance of success when I'm going after it in a rat than I do in a mouse. So I think my success rate of the surgery is likely to be higher. And so we can perform the same kind of studies with a lower number of animals and kind of get at it that way.
Wonderful. Thank you for that explanation. And then just taking a look here, someone just submitted a question, but it's a little bit unclear in the Q&A what is meant. Could you please resubmit with more details, please, or raise your hand to ask your question live? We'll give the person a couple seconds. All right, Tony, we are going to promote you so you can ask your question live. All right, Tony, can you unmute?
Can you hear me now?
Yes, we can hear you.
Okay, could you explain what is meant by, the codes that you use, like N something something, SIC something something.
Yeah, for sure. I'm
I'm
sorry.
I'm a layperson.
Okay. I think I rushed through that a little bit. So SLC9A6 is the gene that's mutated in Christianson syndrome as well as the shake rat. So SLC stands for solute carrier. And let's tie that into, so the protein is NHE6, which is a sodium hydrogen exchanger. So basically, let's just focus on the NHE6 side of things. So really what that means is we're taking a sodium ion. So sodium is Na, so Na plus, so that's where the N comes from. The H is hydrogen, and the E is exchanger. So basically, this is creating it's creating a protein that's very highly expressed in these Purkinje cells in the cerebellum. And so there's a specific compartment inside the cell called the endosome. We don't need to get into that too much. I guess it can get confusing pretty quickly. But basically, when you're exchanging these sodium and hydrogen ions, essentially what you're doing is you're regulating how acidic this compartment of the cell is. So you can think of in terms of you can have acids or bases, and so this particular protein is really important for regulating how acidic this area of the cell is. So basically, when the NHE6 isn't functioning correctly, you end up with too much acidity in this particular intracellular compartment, and that causes a lot of problems kind of downstream from that. Ultimately, it ends up with the cell dying based on the problems that overacidification causes. So when we're talking about NHE6 and SLC9A6, really what we're talking about is a gene that creates a protein that's really important for how acidic part of the cell is in a way that can be fatal to the cell if it's not working properly.
Well, wonderful. Thank you for your explanation, Dr. Anderson, and thank you for your question, Tony. Unfortunately, we are out of time. So if everyone could join me at your individual Zoom screens, thanking Dr. Anderson for sharing his expertise and his research with us. We really appreciate the talk that you gave for us today.
Yeah, thank you so much. Thank you for having me.
And then for everyone who was tuning in and who stayed until the very end, thank you so much for joining us. I hope that you learned something valuable. And wherever you are in the world, good morning, good afternoon, good evening. Have a wonderful rest of your day.